Hello, welcome to Pathagonia. This is Jay. It's been a while, but we're getting back into it. Huge shout out to Dr. Schaberg for his excellent notes. Without him, I'd be more anxious about prepping for boards because my boards is coming up. Okay, so for molecular pathology, there's a quote about molecular pathology from Dr. Juan Rosai. I mean, you can read it yourself, but the last sentence is future discoveries will not be likely made by morphologists ignorant of molecular biologic findings or biologists scornful of morphologic data, but by those willing and capable of integrating them through a team approach. Meaning that molecular pathology is here to stay and is needed to complement our histologic diagnosis. So what are the reasons for using molecular pathology? It helps us end the patient in two main ways. One, neoplasm classification. Increasingly, it is being recognized that many tumors have characteristic underlying molecular alterations. For instance, these alterations are becoming even definitional. This is particularly true in the CNS. For example, while oligodendroglioma was previously defined solely by morphology, like the fried egg appearance, chicken wire vessels, it is now further defined by IDH mutation and 1p19q co-deletion. And then another reason for doing molecular is therapy selection and prognosis. Precision oncology is the molecular profiling of tumors to identify targetable alteration and select specific cancer treatments. The prototypical example of this is treatment of CML, chronic myelogenous leukemia, which has a characteristic BCR-ABL rearrangement with the receptor tyrosine kinase small molecule inhibitor imatinib. This has transformed CML from a death sentence to a mostly chronic disease. Unfortunately, this success of a single type of precision targeting agent achieving near universal durable benefits is mostly unparalleled in solid tumor oncology. So CML is, in a way, an exception. And this is likely because most solid tumors often have additional co-driver and passenger mutations, which are not present in normal early CML. So while these therapies achieve some response in solid tumors, it's often relatively temporary and partial, at least for now. Let's talk about this concept of tumor agnostic therapy. This is treatment based on molecular testing biomarkers rather than tissue type histology. For instance, immunotherapy like the PD-1 antibody pembrolizumab, which is approved in all PD-L1 IHC positive tumors, regardless of the tumor type, mismatch repair deficient, and tumor mutation burden high tumors. Regardless of the tumor, if any of these are present, you're eligible for pembrolizumab. And then you have gene-targeted agents like NTRK, which can be seen in secretory carcinoma of the breast, salivary gland, infantile fibrosarcoma, and in some papillary thyroid cancers as well. BRAF, RET, HER2-directed therapy. This seems to work for some tumor and biomarkers, but not others. For example, NTRK inhibitors seem to work in a wide variety of tumors, while BRAF inhibitors seem to work in BRAF-mutated melanoma and hairy cell leukemia, but not BRAF-mutated colorectal cancer due to the EGFR alternate pathway activation. That's important. One reason why we do BRAF testing and KRAS testing and NRAS testing for colorectal cancer is that's downstream of EGFR. So if those are mutated, EGFR therapy won't work because you have a downstream mutation. Into our high school, college days with the very basics, the central dogma of biology, where DNA becomes RNA, becomes protein. So this is just to dramatically oversimplify. It's not the whole 9 yards, but in a nutshell, DNA encodes through its sequence of 4 different base pairs the genetic information of the body. This is then transcribed into messenger ribonucleic acid, mRNA, which undergoes splicing to remove the non-coding introns. This is then translated into protein, a polypeptide chain composed of amino acids. And each step involves complexes of enzymes, RNA polymerase, DNA polymerase, and etc., that work together in coordination. And there are types of genetic change. So let's talk about mutation and variant. That's any change in the DNA sequence of a cell. That's not to say it could be good, it could be neutral, it could be bad. Often mutation is used in the somatic setting and variant in the germline setting, like Lynch syndrome, BRCA. Somatic mutations are acquired mutations that are not inherited or passed on, mostly studied in tumors. So a somatic mutation, for instance, is paroxysmal nocturnal hemoglobinuria, the PIGA gene. That's an acquired mutation. A germline variation is heritable sequence changes that are in the germ cells and pass between generations. BRCA, Lynch. And then a variant that is expressed in at least 1 to 2% of the population is called a polymorphism. Types of mutations. Missense mutation is a change in one DNA base pair leading to change in one amino acid. It may or may not alter function. An example of this is sickle cell disease, where in the beta globin chain, glutamate is changed into valine. Synonymous mutations. It results in no change in the amino acid sequence. Nonsense mutation. The change in one DNA base pair changes one amino acid to a stop codon, and so there's premature stoppage of the translation. In essence, it's a short protein, and it's often non-functional. Insertions are addition of at least one base to DNA sequence. Deletion, or del, is removal of at least one DNA base pair, and sometimes entire sections and genes. Duplications, sections of DNA copied several times. Frame shift, addition or loss of DNA base pair. This alters the reading frame and alters subsequently translated amino acids and resulting protein, and can also cause a premature stop and often a non-functional protein. Repeat expansion, expansion in the number of times a DNA sequence is repeated. Conversion is a sequence change where nucleotides are replaced by a sequence from elsewhere in the genome. Deletion insertion, or an indel sequence, is where greater than or equal to one nucleotide is replaced by greater than or equal to one nucleotide, essentially a big substitution. And it's important to keep in mind that non-coding mutations also have implications by altering mRNA splicing, promoter function, and microRNA binding sites. So remember to keep that in mind. Let's talk about types of genetic changes. You have the structural variants. These are large changes, often greater than 50 megabases at the subchromosomal level. So examples include translocation, your CML 922, follicular 1418, mantle cell lymphoma 1114. Especially in the hematologic cancers, you can see a lot of translocations. These are chromosomal arrangements between parts of non-homologous chromosomes. The two chromosomes break and the fragments rejoin at breakpoints. Transposition, nucleotides move from one position to another in the genome. It's deletion from one point with an insertion at another point. Inversion, this is a fragment of a chromosome that breaks off and reattaches in the opposite direction. Ploidy, this is gain or loss of the whole complement of chromosomes. You can be hyperdiploid, hypodiploid, for instance, in a partial mole. Oftentimes you get two sets of chromosomes from dad as well as a set of chromosomes from mom. So you're triploidy. It's not just XXY, it's the whole set of chromosomes from 1 to 23. And then different but related is aneuploidy. This is the presence of an abnormal number of chromosomes in a cell that is not a multiple of the haploid state, 23 in humans. It's present in the majority, approximately 70% of cancers. For instance, trisomy 21. It's only the 21 chromosome that has 3. It's not the whole set of 1 to 23 that has an extra chromosome. Epigenetics. These are heritable traits or stable change of cell function that happen without changes to the DNA sequence. For instance, DNA methylation. This is an addition of a methyl group to cytosine, and it occurs in the cytosine guanine-rich areas, the CpG islands. So if it's methylated, then that portion of the DNA is going to typically be silenced for expression. Histone post-translational modifications. These are modifications of specific amino acids on the tails of histone proteins, proteins where DNA wraps around, as you can see in this diagram here. And depending on the modifications, the associated DNA will form closed, non-expressed heterochromatin. And I say heterochromatin to emphasize it's closed, or open, actively transcribed euchromatin to emphasize how it's actively open and transcribed. And so this is a complex with lots of interactions. For example, if you do acetylation, that lowers the positive charge on histones. That decreases the binding to the negatively charged DNA, so it's easier to access for the transcription factors and RNA polymerase, and then henceforth more transcription. Methylation, this attracts enzymes that further methylate DNA, leading to decreased gene expression. And here is a micrograph of DNA where you can see the heterochromatin, and then you have the euchromatin where it's open, expressed, it's less dark and condensed. And here's the H&E correlate of the electron microscopy where you see the nucleolus, the heterochromatin, and the euchromatin. And this will be talked about later, but if there are any mutations in the histone proteins themselves, sometimes the tumors will be rhabdoid, and they usually have poor prognosis. But that was just a preview. We will talk about it further along.
All right, let's talk about how mutations cause tumors. So tumorigenesis is often a multi-step process where cells acquire successive mutations. And you want to focus on 2 main carcinogenic gene changes. 1, increased proto-oncogene function. Think a car gas pedal getting stuck down, so it's just going to accelerate no matter what. 2, loss of tumor suppressor function. Think cutting the brake lines on a car and can't stop. The tumor suppressor function often needs the 2-hit mutations. Proto-oncogene often needs just 1 hit. And either one results in uncontrolled cell growth and failure to respond to normal signals that lead to cell death. Now, oncogenes. So proto-oncogenes are genes involved in normal cell growth. They're normal. It's oncogenes that are the mutated version of a proto-oncogene that increases expression or activity of proto-oncogene through a gain of function. And so you have increased growth, cell division, and inhibition of cell death. And it's mostly transcription factors. You only need 1 copy to cause the disease state, as mentioned before, and it acts in a dominant fashion. If a tumor is profoundly dependent on a specific mutation protein, it is called an oncogene addiction, and it's a great target for therapy. Think imatinib for BCR-ABL. Common examples are point mutations, deletions, and insertions that may lead to increased transcription. Amplification leading to increased transcriptions. Translocation brings proto-oncogene to a site with increased expression, leading to increased transcriptions. Now, tumor suppressors, the brakes of the car, so to speak, not working. The tumor suppressors encode proteins that repress the cell cycle, quote-unquote growth, and promote apoptosis or cell death, leading to a loss of function due to the mutation, and it no longer inhibits uncontrolled growth. Since we are diploid, meaning we have 2 copies of every gene, 1 from mom, 1 from dad, you need 2 hits to deactivate both copies. and it acts in a recessive fashion. Common tumor suppressor roles include DNA repair enzymes, repression of genes essential for continuation of cell cycle, coupling of cell cycle to the presence of DNA damage with pro-apoptotic signaling if DNA damage cannot be repaired, cell adhesion molecules that help prevent metastasis. Driver versus passenger mutations. They often occur simultaneously in the same tumor genome. And driver mutations are those that alter the function of cancer genes and directly contribute to development and progression. It's tightly clustered within cancer genes and more relied upon by cancer cells. Often a small subset of overall mutation and often mutually exclusive. It provides a selective advantage, and it's more often than not the targets of therapy like EGFR with erlotinib for lung carcinoma or EML4 ALK. Passenger mutations, these are mutations that are less important for growth and survival. So it's more neutral and is often more numerous, random, and sprinkled throughout the cancer genome. But it's neutral at first, but they may provide a selective advantage in the setting of therapy. Genetics leading to morphology. Broadly speaking, you can get a hint as to the genetics of a tumor by the basic morphology, and I find that so fascinating. So, for instance, the more genetic changes a tumor has, often it's more pleomorphic, heterogeneous, and atypical. Tumors with the recurrent driver mutation fusions often appear relatively bland and homogeneous or monotonous, with all cells having similar genetics. For instance, dermatofibromatosarcoma protuberans, DFSP, is translocation 1722 PDGFRB:1A1, and that looks more monotonous than just even your dermatofibroma in general. So complex genetics, overall, this is more common. Examples include UPS or undifferentiated pleomorphic sarcoma, myxofibrosarcoma, and high-grade carcinomas. And there are lots of genetic changes, often with many passengers and subclones. And here's an example Dr. Schaberg provides with a karyotype of leukemia, and you can appreciate the immense aneuploidy. And here's a picture of just how significant pleomorphic and heterogeneous tumors with complex genetics are. In contrast, simple genetics, these are overall less common. They're most common in sarcomas, around 20%, and hematolymphoid tumors. Synovial sarcoma, X18, infantile fibrosarcoma, secretory carcinoma, both are 1215 ETV6 and NTRK3. Adenoid cystic carcinoma is 6-9 MIB NFIP. And fewer genetic changes, often one main driver mutation, frequently a fusion protein. And so here's an example of synovial sarcoma with the X18 translocation. This being said, these tumors with simple genetics are very testable on the boards. So please remember these, have these translocations in your memory, because it'll make boards, you'll save a lot of time on boards when they ask you the question. It's just, you know it, and it'll save you time. So here's an example of a monotonous morphology with more bland cytology in tumors with simple monotonous genetics. So let's talk about tumor suppressors, not oncogenes. So tumor suppressors help with the cell cycle checkpoint, and ATM will activate P53 and will help with MDM2. MDM2, like liposarcoma, MDM2 amplification, that will inhibit P53 and will inhibit the downstream effects of P53 towards P21, which will stop the growth from G2 to the M phase. And it'll also inhibit retinoblastoma. Also, P16, the one that we get for HSIL in the cervical cervix, that will inhibit cyclin D1 CDK4 complex. And what this complex does is it phosphorylates retinoblastoma and makes it inactive. So by inhibiting this complex, P16
prevents the does not allow retinoblastoma to get inactivated. So an important point I want to point out is, HPV, the virus, has the E7 protein that ultimately disrupts the retinoblastoma pathway. But if there's other oncogenic stresses that inhibit the retinoblastoma pathway, you can imagine that tumor would be P16 positive. Like some oropharyngeal tumors, they're P16 positive, but that doesn't necessarily mean it's HPV driven. And that's why you're encouraged to get a HPV-ish when an oropharyngeal tumor is P16 positive. Okay, TP53, the guardian of the genome. So TP53 is the gene, and the protein is P53, just minus the T. It is the most commonly mutated gene in human cancer, approximately 70% of cancers. And P53 is activated by stresses like DNA damage and causes G1 arrest, induces expression of DNA repair enzymes, and induces apoptosis if repair fails, through many complex pathways and interactions. So a person with a germline mutation of P53 is Li-Fraumeni syndrome. And so you just need, one of your cells needs just one hit of P53 because you already have a mutated P53. And this can be inactive. P53 can be inactivated by viruses like the E6 protein in HPV, causing carcinogenesis. The way I like to remember how E6 affects P53 is right after 5 is 6. So it's like 5, 6. And then E7, then I know it's retinoblastoma. MDM2 inhibits P53, so its amplification, as mentioned before, in liposarcomas lead to loss of P53 function. P53 IHC is a quick and easy surrogate marker for TP53 mutation in most circumstances. Very good specificity, reasonable sensitivity. The most common pattern is diffuse strong nuclear positivity, overexpression, involving at least 80% of the tumor cells, but usually almost 100%. Wild type staining is characterized by an admixture of negative cells, weakly and strongly positive cells. Notably, some splice site mutations or truncating mutations can result in detectable but non-functional P53 protein, yielding a normal wild-type staining pattern. So that's very tricky. Interpretation of complete loss requires good internal controls, lymphocytes and fibroblasts to accurately interpret. So P53 in the GYN system, the well-known is high-grade serous carcinoma of the ovary or serous carcinoma of the endometrium. They're both P53 driven. Endometrioid carcinoma, if you got a P53, it shouldn't be P53 overexpressed or lost completely or cytoplasmic. It should just be wild type. So I'll leave this chart for your review, but this wild type is what P53 wild type looks like right here. Overexpressed, you can see the diffuse positivity, complete loss. The things that are positive are the background lymphocytes and fibroblasts, as acts as an internal control, and cytoplasmic. One, known as the governor of the cell cycle. The gene is retinoblastoma 1, the protein is retinoblastoma. It's a key negative regulator of the cell cycle. It's directly or indirectly inactivated in most human cancers. It regulates the G1S checkpoint, and cells must pass to replicate DNA. It's the first tumor suppressor gene to be discovered. It's the prototype for family cancer genes after found to be mutated in familial retinoblastoma, in which one germline allele is inactive at birth, and the second hit leads to cancer. Retinoblastoma mutations are common in small cell carcinoma, breast, and bladder, and functionally inactivated by E7 protein in HPV to cause cervical squamous cell carcinoma. And the detection, you can do an IHC and you look for loss of staining. Here you see loss in small cell carcinoma with an internal control of maybe lymphocytes here. And you could do next-gen sequencing as well. Cyclin D1, mutations
and amplifications lead to retinoblastoma phosphorylation, which leads to retinoblastoma inactivation, and there's no checkpoint. The genes are overexpressed in many breast, esophagus, liver cancers, as well as lymphomas and plasma cell neoplasms. By IHC, it's the staining pattern. You'll see retinoblastoma loss, P16 block positive, and cyclin D1 low loss. And then just a key note about cyclin D1 in particular, because this was on my practice boards, but another name for it where they might trick you by saying this is BCL1. BCL1 is cyclin D1, so just keep that in mind. CDK4, cyclin-dependent kinase, which helps regulate the cell cycle. Mutations and amplifications lead to retinoblastoma phosphorylation, retinoblastoma inactivation. There's no checkpoint. And then amplifications common in melanomas, sarcomas, and glioblastomas. CDKN2A, this one's particularly more well known. So the gene is CDKN2A and the protein is P16. So it's a tumor suppressor, a CDK inhibitor, frequently disabled in cancer by mutation or gene silencing. In many cancers, including HPV-mediated squamous cell carcinoma, high-grade serous carcinoma, small cell carcinoma, liposarcoma, abnormally accumulates, and this leads to a diffuse strong block positive staining by IHC. I want to emphasize here that P16 staining does not necessarily mean HPV infection. HPV can lead to overexpression of P16, but there are other oncogenic stresses that lead to P16 overexpression. For instance, liposarcoma that's diffusely P16 expressed, that's not driven by HPV. It's driven by some other oncogenic stress that has the same common endpoint of overexpression of P16. So I hope that helps. Notably, this is only a surrogate often for other abnormalities like mentioned, like HPV, which can be investigated separately by doing a reflex HPV-ish. CDKN2A deletion is also seen in mesothelioma and can be detected by FISH and can be helpful in differentiating benign from malignant mesothelial proliferations. ALK, the prior name was anaplastic lymphoma receptor tyrosine kinase. So this encodes ALK receptor tyrosine kinase. It binds the ligand and it induces homodimerization and leads to activation of multiple pathways, RAS, JAK, PI3K, all leading to cell proliferation and survival. There are three types of alterations: ALK translocations, where the fusion proteins lead to ligand-independent constant activation, usually good prognosis. Common examples include the NPM1 ALK in ALCL, or anaplastic large cell lymphoma. It's translocation 2-5, I believe. EML4 ALK in lung cancer, mutually exclusive of EGFR or KRAS, and highly sensitive to ALK inhibitors like crizotinib. ALK mutations, many reported, usually confer resistance to tyrosine kinase inhibitors. ALK amplifications are present in many tumor types. And alteration detection, IHC often used for diagnostic purposes. ALK positive ALCL and inflammatory myofibroblastic tumor, although only around 70% of IMTs are positive for ALK. So you also want to look for the morphology of IMTs. For targeted therapy, FISH or NGS is often done, which is more specific.
So the RAS pathway, remember RAS is an oncogene. The other ones we have mentioned were tumor suppressors. So RAS, the downstream effect is it activates transcription and leads to production of proteins like Myc, and that leads to cell cycle progression and growth, leading to cancer. 3 RAS genes you should know are KRAS, HRAS, and NRAS. It is the most commonly mutated oncogenes in human tumors. Remember, P53, APC, these other well-known genes, they're tumor suppressors. Activated RAS stimulates downstream regulators of proliferation, leading to uncontrolled growth, as we have mentioned. KRAS mutations, most commonly activated by point mutations. either at GTP binding or enzymatic region. Either way, it doesn't break down GTP and stays activated instead of normal cycling on and off. I remember that being one of my practice board questions. What's the most frequent way KRAS gets mutated? Well, about 30% of all tumors, and it's higher in GI and lung. For example, greater than 90% of pancreatic ductal carcinomas, approximately 50% of colorectal. In lung, it's associated with smoking and confers a worse prognosis. And it confers a resistance to anti-EGFR therapy. So that's why if you have colon cancer, you want to check for the KRAS mutation status, because if they do have the mutation, then you know that they would not respond to these EGFR therapies. KRAS is more commonly seen in metastatic cancers, leading to a worse prognosis. KRAS amplifications can be seen in a variety of cancers, including colorectal, and you can detect this with NGS. BRAF. This is serine threonine kinase in the RAF-ERK-MAP kinase pathway. It is usually activated by somatic missense mutations, leading to it being constantly active. And the most common, as you know, is BRAF V600E. It is the most common oncogenic driver in melanoma, accounting for approximately 60%. BRAF V600E mutations are also seen in papillary thyroid carcinoma, around 70%, hairy cell leukemia, nearly 100%, Langerhans cell histiocytosis, around 60%, colorectal cancer, around 10%, usually sporadic MSI high, meaning it's not Lynch, and non-small cell lung carcinoma, around 5%. And you can detect this usually with NGS. There is a BRAF V600E mutation-specific IHC that shows cytoplasmic staining if positive, as you can see here with diffuse cytoplasmic staining. Of note, some normal tissue, like cilia, can show some degree of staining with the BRAF V600E IHC. The therapy for BRAF mutations, you can give BRAF inhibitors like vemurafenib, but it often develops resistance. Let's talk about EGFR, epidermal growth factor receptor. This is an oncoprotein that functions as a tyrosine kinase in the EGFR pathway, leading to cell proliferation. It's altered in 30% of epithelial tumors, and it correlates with tumor progression and resistance to chemotherapy. EGFR mutation is found in approximately 15% of non-small cell lung carcinoma with adenocarcinoma morphology, and approximately 40% in Asia. It's more common in female non-smokers. Remember, your BRAF and KRAS are the ones that are generally seen in smokers. EGFR mutations in these lung cancers have better prognosis. The most activating mutations are in exons 18 to 21, and it leads them to be sensitive to EGFR inhibitors like erlotinib and gefitinib. T790M in exon 20 mutations confer resistance to inhibitors and can develop pre or post inhibitor therapy, and then in that case you can give them osimertinib. EGFR amplifications you can see in non-small cell lung carcinoma, often squamous cell carcinoma, glioblastoma, and others. And you can detect the EGFR mutations through next-gen sequencing, and the therapy is EGFR small molecule inhibitors like erlotinib and gefitinib, used in advanced metastatic non-small cell lung carcinoma. Anti-EGFR monoclonal antibody cetuximab is used in metastatic colorectal cancer. However, if there is RAS or BRAF mutations, then it will be resistant to cetuximab. HER2. The gene is ERBB2, protein is HER2/neu, and it's part of the EGFR family, and it's a tyrosine kinase inhibitor involved in growth and signaling. HER2 amplification and overexpression. It's most common in breast and gastroesophageal carcinoma, and a smaller proportion of GYN salivary lung. HER2 mutations are less common than amplification. It's seen in lung, breast, and other tumors. And you can treat with trastuzumab, Herceptin. It's a monoclonal antibody directed against HER2. I always get confused with trastuzumab, Herceptin, and tamoxifen. They both start with a T, but tamoxifen is anti-estrogen, and trastuzumab is anti-HER2. Trastuzumab deruxtecan and HER2. This is a trastuzumab conjugated with the chemotherapy drug deruxtecan. And you can detect with IHC and FISH. HER2 amplification overexpression can be detected by IHC, with equivocal cases being reflex to FISH. So this is an example of zero negative staining, and Dr. Schaberg provides us kind of an analogy of an eraser. No staining is observed. There's a faint, barely perceptible membrane staining, like you can do with a pencil with a thin point. That's 1+ negative. If you can make it with you can make the borders out with a pen. That's weak to moderate membrane staining, and it's 2+ equivocal. And 3+ positive is when you make the membrane staining with a Sharpie, and a strong, complete membrane staining. So RET encodes transmembrane receptor tyrosine kinase, leads to activations of many pathways, including RAS, MAPK, and PI3K, AKT, and leading to cell growth and proliferation. RET fusions results in ligand-independent activation. Approximately 1 to 2% of non-small cell lung carcinoma and 10 to 20% of papillary thyroid carcinomas. And you can detect with NGS or FISH. And RET mutations, these are germline mutations in MEN2A and 2B, and somatic mutations in sporadic medullary thyroid carcinoma, and you can detect with NGS. And several multi-kinase inhibitors like sunitinib have activity with both alterations. Let's talk about MET. The gene is MET, the protein is HGFR. So receptor tyrosine kinase, and it can lead to various pathways, including the PI3K, AKT, and the RAS MAP, leading to ultimately self-proliferation. MET overexpression through amplification or epigenetic changes is the most common alteration, particularly in lung cancer. It often correlates with poor prognosis in many tumor types. MET gene amplification can be secondary to treatment with anti-EGFR tyrosine kinase inhibitors. In particular, MET mutations, the exon 14 skipping mutations, which is important to know, can be seen in non-small cell lung carcinoma and have specific targeting drugs, capmatinib and tepotinib. You can detect this with FISH or NGS, and you can treat with a MET inhibitor, but know that you often can develop the resistance to these. Pten, which is associated, I always think with caudins. When I think of Pten, automatically caudins. But not necessarily it's always caudins. It's a tumor suppressor, virtually ubiquitously expressed, and it regulates cell proliferation and apoptosis through inhibition of AKT via the PI3K AKT pathway, and inactivation leads to cell proliferation. Germline mutations, it leads to the Pten hamartoma tumor syndrome or caudins, where you get trichomomas, endometrial carcinomas, breast cancers, follicular thyroid carcinomas. Mutations are frequently seen in endometrioid carcinoma and useful diagnostically in case you're split between that and serous carcinoma, prostate cancer, glioblastomas. And to detect, you could do IHC for diagnostic purposes, but it's not high enough sensitivity or specificity, around 80%, to be the absolute surrogate marker. So you may need NGS in some circumstances. Remember, this is normal. What's abnormal is Pten loss. Myc. This functions mainly by activating transcription of other genes, including those that drive the cell cycle. Dysregulation promotes tumorigenesis by simultaneously promoting the progression of the cell cycle and enhancing alterations in metabolism that support cell growth. Translocation of Myc with immunoglobulin gene loci in Burkitt's lymphoma, so its translocation, common one is 814, and other lymphomas like DLBCLs and leukemias. And it's also amplified in breast, colon, lung, and neuroblastoma, and Myc.
And you can detect Myc translocations often confirmed by fish break-apart assay. And although the IHC exists and protein expression is often upregulated with translocation, this is not considered an adequate surrogate marker. ROS1, this encodes cell surface receptor tyrosine kinase and activation signals through multiple pathways like MAPK, PI3K, leading to cell growth and proliferation. It's involved in fusion proteins with a variety of partners and signaling in the absence of a ligand, and fusions in 1 to 2% of non-small cell lung carcinomas. Classically in young female never smokers with adenocarcinoma, and you treat with crizotinib. That's important to know. ROS1 mutation, non-small cell lung cancer, treat with crizotinib. Alteration you can detect with NGS or FISH. WNT pathway. So I'll leave this a helpful diagram that Dr. Schaberg has for your reference. But in general, in normal resting cells, APC binds to beta-catenin and leads to its destruction. And in the WNT stimulated or mutated tumors, beta-catenin is released, non-degraded, and translocates to the nucleus, proliferation. And that is why when you get a beta-catenin and it has nuclear expression, that is why it's abnormal. So let's just briefly talk about the WNT pathway. E-cadherin maintains cell contact inhibition in epithelium, which can be lost in malignancy. The normal cells stop proliferating when they lose contact with each other, but cancer cells keep growing and they look discohesive. In this case, it's an intact E-cadherin, but this is lost. And it's lost in lobular breast cancer. It can be diminished in lobular breast cancer and diffuse gastric cancer. Both are driven by that CDH1. And you detect with IHC for ECAD. And APC, tumor suppressor, is discovered in adenomatous polyposis coli syndrome, and it encodes a cytoplasmic protein whose main function is to promote degradation of beta-catenin. Patients with APC germline mutations develop multiple adenomatous polyps in the colon and have a high rate of colon cancer. You already have a one hit in a setting that has a lot of cell turnover. So it's only a matter of time that one of the colonic cells has two hits and develops into tumor and cancer. Beta-catenin CTNNB1 gene has two main roles. One, it binds E-cadherin and is involved in cell adhesion and contact inhibition. Two is when translocated to the nucleus, it binds transcription factors and coactivates genes that promote the cell cycle and proliferation. Detection, you can do IHC for beta-catenin and it shows a membranous staining in many cells. With the WNT pathway mutations such as CTNNB1, nuclear beta-catenin staining can be seen. Warning, it can be very focal, so you gotta look thoroughly, sometimes even like at 40x, which can be useful diagnostically in tumors such as desmoid fibromatosis, nasopharyngeal angiofibromas, and certain hepatic adenomas, specifically like the beta-catenin one. IHC shows very good specificity, meaning if it's positive, then you do have it, but only 80% sensitivity, meaning like even if it's negative, you can still have it 20% of the times. So NGS may be necessary in negative cases if it's important to confirm a mutation. So Dr. Schaberg has these wonderful diagrams highlighting the point where you can see the nuclear beta-catenin, and sometimes you have to look closely because it's surrounded by cytoplasmic blushing. I mean, this is a great case, but that's not always the case. Like for this case, you really have to look closely to see that nuclear beta-catenin. These are chromatin remodeling complexes. They regulate nucleosome positioning and thus alter transcription and also likely play a role in DNA repair. I get scared of these because the prognosis here are pretty bad. They have a complicated macromolecular assemblies consisting of many subunits, and nearly 25% of all cancers harbor aberrations in one or more of these genes, and it's linked to a worse prognosis across several cancer types, but not universal, but particularly true for SMARCB1, leading to the protein INI1. So some of the subunits that are we should know are the SMARCB1. That protein is INI1, and it's mutated in epithelioid sarcoma, atypical teratoid rhabdoid tumor, or ATRT, rhabdoid tumor, all aggressive. I mean, if there's a tumor that has the name rhabdoid, and you're not sure what mutation it has on the exam, think SMARCB1. SMARCA4 protein is BRG1. The way I remember the difference between SMARCB1 and SMARCA4 in terms of the protein is SMARCA4, the next letter next to A is B, so that's BRG1. And this is mutated in SMARCA4 undifferentiated thoracic tumors, small cell carcinoma of the ovary hypercalcemic type, and undifferentiated endometrial carcinoma, all aggressive. ARID1A is mutated frequently in clear cell and endometrioid carcinomas of the gyne tract. PBRM1 is mutated often in clear cell renal cell carcinoma. And SS18, the SS18 SSX fusion is found in synovial sarcoma, which is an X18 translocation. Detection, it varies based on the subunit, but there is IHC that exists for INI1, BRG1, ARID1A, where loss is indicative of mutation. SS18 SSX fusion was historically tested for by FISH. Now there is an IHC stain for the SS18 SSX chimeric protein. That is a useful tool for synovial sarcoma differential diagnosis, but unusual immunophenotypes should trigger molecular genetic testing. The genetics leading to morphology leading to behavior. So particularly for SMARCB1, protein INI1, and SMARCA4, the protein is BRG1 deficient tumors. These tumors are often very aggressive and often have a similar morphologic appearance with poorly differentiated, somewhat discohesive rhabdoid to basaloid cells with vesicular chromatin, prominent nucleoli, and abundant necrosis. These tumors show variable keratin staining and sometimes stain with CD34. If you ever have a bad looking tumor with morphology like this, consider getting INI1 and or BRG1 IHC to evaluate for one of these tumors, as they can be seen in most organs. So Dr. Schaberg provides an H&E of this atypical teratoid rhabdoid tumor. This is a small cell carcinoma of the ovary of hypercalcemic type, and this is just a rhabdoid tumor. And here's an example of a loss of SMARCB1 INI1 by IHC. EWSR1. This was discovered in Ewing sarcoma. Hence, just like DOG1 is discovered on GIST, EWSR1 is Ewing sarcoma breakpoint region 1, EWSR1. It's involved in RNA binding, transcription, and RNA metabolism. The role in cancer cell progression is still unclear, although it may play a critical role in DNA damage, response, and cell division. Nevertheless, fusion oncogenes can act as aberrant transcription factors, deregulating genes involved in tumorigenesis. There is significant fusion gene promiscuity, as EWSR1 can fuse with many other genes. So this is a quote-unquote dirty gene. One interesting aspect of this promiscuity is that identical fusion genes are present in tumor types that have entirely different histologic features and biologic potential. It's possible that tumorigenesis depends on the differentiation state of the cell and its cellular compartment. For instance, angiomatoid fibrocystocytoma and clear cell sarcoma can both have EWSR1 CREB1 fusions, the former having intermediate behavior, less than 5% metastasized, and then the latter, clear cell sarcoma, is malignant, has poor prognosis. Clear cell sarcoma has packets of cells with clear cytoplasm and with prominent nucleoli, and then angiomatoid fibrocystocytoma are kind of somewhat spindle to epithelioid cells, a little bit more basophilic appearing. They have more blood vessels, and they have this characteristic lymphoid cuff. And then you have your Ewing sarcoma, the prototypical small round blue cell tumor. Dr. Schaberg provides an excellent chart detailing the different fusions that EWSR1 can have. I leave this here for your review, but just highlighting EWSR1 FLE1, that's the most common fusion in Ewing's sarcoma. EWSR1 CREB3L1 is low-grade fibromyxoid sarcoma, and somewhat related to that entity is the sclerosing epithelioid fibrosarcoma, which also has a EWSR1 CREB3-1. You have your EWSR1 WT1, which is desmoplastic small round cell tumor, and EWSR1 NR4A3 for extraskeletal myxoid chondrosarcoma. To detect, you could do FISH, RNA, NGS, or RT-PCR panel. If Dr. Schaberg is confident of the diagnosis on H&E, he usually gets FISH as it's faster, but if he's unsure, he will do NGS or RT-PCR to identify the fusion partner, which can be diagnostically helpful. All right, KIT, aka c-KIT, aka CD117. This is a receptor tyrosine kinase. Homodimerization induces a signaling cascade that has a role in cell survival, proliferation, and differentiation. Activating, again, I emphasize activating mutations are found in many tumors, including GIST, systemic mastocytosis, approximately 75%, AML approximately 25%, and a subset of mucosal melanomas, approximately 20%. You treat with receptor tyrosine kinase small molecule inhibitors like imatinib with often good response. The specific exon mutation often impacts prognosis and RTK inhibitor sensitivity, so tumors are often sequenced to determine therapy and prognosis. To detect this, you can do NGS. There is no correlation between immunohistochemical expression of CD117 and KIT mutations. For instance, if a suspected GIST stains for CD117, it doesn't necessarily mean that there is an activating KIT mutation. It can be possible that it has an SDH mutation, and which is associated with the Carney triad, Carney-Stratakis syndrome. And so just because CD117 IHC is positive, you still want to make sure that it has a KIT mutation is the point. PDGFRA, platelet-derived growth factor receptor alpha. It's a receptor tyrosine kinase in the SAM family as KIT. PDGFRA mutations found in GIST, approximately 10%. Some, like D842V, indicate resistance to imatinib, while others do not. PDGFRA fusions seen in various hematolymphoid tumors. Most common fusion is FIP1L1 PDGFRA, which most commonly presents as chronic eosinophilic leukemia, the response to receptor tyrosine kinase inhibitors like imatinib. You can detect mutations by NGS, fusions by FISH or RNA, NGS, or RT-PCR. FGFR, fibroblast growth factor receptors. These are a family of receptor tyrosine kinases that bind fibroblast growth factors. Downstream signaling via the MAP kinase, JAK-STAT, and PI3K-AKT pathways. Alterations are most common in FGFR1, followed by FGFR2, 3, and 4, respectively. FGFR amplification seen in approximately 20% of lung SCC, also some urothelial breast. FGFR mutation, many different point mutations described in various tumors. FGFR fusion seen in some cholangiocarcinomas, gliomas, and hematolymphoid malignancies. And it can be treated with multi-target tyrosine kinase inhibitors or FGFR selective tyrosine kinase inhibitors. NTRK, neurotrophic receptor tyrosine kinases. These activate the downstream MAPK pathway. There's 3, NTRK 1, 2, and 3. NTRK fusions, these are seen in a variety of tumors of soft tissue, thyroid, salivary, gyne, gliomas, etc. And it's important for us to know on boards, especially the most common, the ETV6 NTRK 3, which is congenital mesoblastic nephroma, infantile fibrosarcoma, secretory carcinoma. It's the translocation 1215, and NTRK rearranged solid tumors, regardless of tumor type, usually respond to treatment with a targeted inhibitor of the NTRK tyrosine kinases, larotrectinib and entrectinib. So these are tumor agnostic therapies. Regardless of the tumor type, if you have an NTRK fusion or rearrangement, then you're eligible for these like larotrectinib. NTRK mutations can be seen in a variety of tumors and can contribute to drug resistance. It's very rare types of papillary thyroid carcinoma can have NTRK fusions or mutations. Detection, you can do a pan-TRK IHC, shows some utility as a diagnostic and surrogate marker for NTRK screening. However, physiologic or nonspecific expression may lead to false positive results. So RNA testing is often required to confirm. Let's talk about IDH1/2, or isocitrate dehydrogenase 1 and 2. These are enzymes involved in Krebs cycle and metabolism. And IDH mutations, it's present in the vast majority of astrocytomas and oligodendrogliomas, usually R132H. And that's one way you can distinguish glioblastoma from these other two glial tumors, astrocytomas and oligodendrogliomas, because there will not be IDH mutations for glioblastoma typically. Also, chondrosarcomas, approximately 50%, cholangiocarcinomas, 20%, hematolymphoid tumors, 10%, AML, and 20% angioimmunoblastic T-cell lymphoma. Inhibitors of mutant IDH1/2 enzymes are beginning to be used, like vorasidenib for therapeutic uses. And detections, mutation-specific antibodies exist for IDH1R132H, which is the most common mutation in gliomas and works well. Something to note that IDH1/2 mutation leads to an oncometabolite. So if it's present in brain tumors, that means there's an IDH mutation. And so it could be astrocytomas or oligodendrogliomas. If there's no IDH staining, then that's wild type. And that suggests glioblastoma if the location and other factors are right for it. Genetic analysis of IDH1/2 genes by sequencing is recommended for diffuse gliomas when IHC for IDH1R132H is negative. Microsatellite instability / mismatch repair deficiency. So DNA mismatch repair is a tumor suppressor system for recognizing and repairing errors that can arise during DNA replication and recombination, as well as repairing some forms of DNA damage. If these genes are mutant loss deficient, that leads to defective DNA repair, and tons of mutations accumulate, quote unquote hypermutated, and gain or loss of an entire short tandem repeat microsatellite fragments, leading to microsatellite instability, or what we call MSI high. Deficiency in MMR, shorthand is DMMR, is seen frequently in colorectal cancer, approximately 15%, endometrioid carcinoma, 25%, other GI adenocarcinomas, and sebaceous carcinomas. Of the colorectal cancers that are approximately 15% deficient in MMR, I want to emphasize something, and that Dr. Schaberg does too. So it can be germline, Lynch syndrome, that's less common. I've heard one study, it's like 3% of the 15%. And then sporadic is more common, approximately 12% from that paper I read. In colon, sporadic cases are often due to MLH1 promoter hypermethylation, leading to epigenetic silencing of MLH1, leading to DMMR. So if there is DMMR, the more likely statistically is that it's going to be to sporadic change. Detection, universal screening of all new colorectal carcinomas and endometrioid carcinomas. You can do via IHC and or PCR. Most institutions do IHC first, given its ease and availability. Both methods have around 90% sensitivity for identifying Lynch syndrome. Intact nuclear expression of MLH1, MSH2, MSH6, PMS2 leads to an MMR proficient tumor. And loss of nuclear staining with positive internal control with the lymphocytes in the lamina propria here, but the loss in the colonic epithelium, that's MMR deficiency. And to identify Lynch syndrome cases, deficient tumors must undergo MLH1 promoter methylation testing. If present, methylation indicates a sporadic tumor. In colon cancer, BRAF mutations correlate very closely with MLH1 promoter methylation. So BRAF testing has become routine before MLH1 methylation testing, as it is technically less demanding / costly. Generally, MSI high DMMR tumors have lots of mutations, cytologically higher grade, lots of neoantigens presented to the immune system. So you'll see a lot of lymphocytic infiltrates, and that leads to a better prognosis. Note, DNA polymerase, POL-E, and delta mutations can result with a similar clinical phenotype, and these also have a really great prognosis. Therapy implications: automatically approved for anti-PD-L1 therapy, pembrolizumab. Tumor mutation burden, TMB. This is calculated by counting the number of likely somatic coding mutations within a proportion of the tumor genome. Although threshold for a high TMB score varies between panels, TMB high is generally defined as greater than or equal to 10 mutations per megabase, and D, MMR, and polymutated tumors have high TMB. And this is very important for us to know, especially as molecular keeps on gaining ground. Regardless of tissue type. So it's tumor agnostic. TMB high tumors all qualify for immunotherapy for pembrolizumab. Let's talk about histone mutations modifications. Protruding from the nucleosome, which are DNA and histones, are the terminal tails of the histone proteins, which can undergo post-translational modifications and/or mutations. And this can impact gene expression by 1, altering the charge of histones and their attraction to negatively charged DNA, and 2, altering binding of other proteins. Common post-translational changes include methylation, which silences the gene expression, acetylation, which opens up the area for gene expression, phosphorylation, and ubiquitination. Specific histone modifications found in tumors that are relevant to us are H3K27ME3. This is the definition of the pediatric infiltrated midline glioma, known as diffuse midline glioma, H3K27 altered, aka diffuse intrinsic pontine glioma, and it shows loss of the methyl group on H3K27 due to a mutation that replaces the lysine with a methionine. There is an accompanying IHC stain, H3P-K28ME3 or K27ME3, which shows loss. Malignant peripheral nerve sheath tumors, you see them in especially patients with NF1, are characterized by many mutations, including NF1, CDKN2A, and/or CDKN2B deletions, and inactivation of SUS12 or ED. PRC2 mediates the deposition of H3P-K28ME3, an important repressive mark that plays a critical role in cellular differentiation. Inactivation of PRC2 leads to a complete global loss of H3P-K28ME3 in tumor cells, which can be demonstrated by IHC. About 80% of conventional high-grade MPNSTs show loss of H3K27ME3. There's H3G34, which is the definition of diffuse hemispheric glioma, H3G34 mutant, an infiltrative glioma involving the cerebral hemisphere of kids. Missense mutations of the H33A gene results in one of several substitutions on the histone H3 protein, usually G34R, and you can detect this by NGS. Now, HG34W, almost all, greater than 95% of giant cell tumors of bone harbor the pathogenic H33A or H3F3A gene mutations, approximately 90% of which are H3G34W mutated, and this can be detected by IHC. Other mutations can also be tested for by NGS. And H3K36M, the vast majority of chondroblastomas harbor a P-lysine 36 methionine substitution in one of the genes that encode H3.3. This inhibits the H3K36 methyltransferases, which results in reduced global methylation. And IHC using an antibody against H3.3b or antibody against H3F3b K36M shows diffuse nuclear expression in greater than 96% of the cases. DNA methylation, epigenetic modifications of DNA by methylation of cytosine nucleotides. Cancer cells show global hypomethylation and selective promoter localized hypermethylation. Genome-wide hypomethylation leads to chromosomal instability, increased copy number of oncogenes, and decreased copy of tumor suppressors, leading to carcinogenesis. Methylation of promoter DNA leads to decreased gene transcription and expression. It attracts proteins that cause chromatin condensation and block transcription. And you can imagine if this is the promoter of a tumor suppressor gene, that's going to lead to cancer because you're inactivating the brakes, so to speak. The methylation of specific gene promoters has prognostic significance. Promoter methylation of MGMT, which is a DNA repair gene in high-grade gliomas, predicts response to alkylating agents like temozolomide. Methylation profiling can be used in classifying tumors and will likely be of increasing importance given its ability to identify new cancer classes and to consolidate histologically disparate cancers, particularly of the CNS and soft tissue. In fact, the NIH has a large methylscape analysis project and will perform free analysis of any CNS or soft tissue tumors sent to them.
Let's talk about common molecular techniques, or cytogenetics. It allows to look at the entire karyotype. It's low resolution. You can only look at the chromosome level changes only, 5 to 10 megabases. The examples of findings include aneuploidy, large chromosome gain/loss, translocations. It requires living cells and is relatively slow. It is useful, for instance, your leukemias, your B acute lymphoblastic leukemia and lymphoma. You were able to see if it's hypodiploid, if it's hyperdiploid, if it has a translocation 411. And for cytogenetics, you grow the cells in culture, you add colchicine, which causes a metaphase arrest. That's when all the chromosomes are aligned in the middle of the cell. And then cells are dropped on a glass slide and stained to reveal a band pattern. In this case, you can see a balanced reciprocal translocation where you have your Philadelphia chromosome 922, most often seen in CML. FISH, or fluorescence in situ hybridization. This is higher resolution, 40 kilobases, but limited scope of analysis. You only evaluate areas identified by sequence-specific probes. It can be performed on formalin-fixed, paraffin-embedded smears, and it's relatively fast, 1 to 2 days, and you can evaluate multiple cells. The fluorescently labeled probe is specific to a DNA target sequence, and you incubate to allow it to hybridize to cell DNA, and you apply DAPI counterstain to see the DNA, and you view under a fluorescent microscope. Examples of findings include fusions and amplifications. So here we see a translocation of EWS, and here we see amplification of a certain gene. For instance, it could be a Myc gene amplification, it could be HER2 amplification. And 3 probe strategies. One is for chromosome and gene number. It detects the presence or absence of specific chromosomes/genes. Fusion probes. If 2 genes involved in a translocation are known, gene number 1 is bracketed by red probes and gene number 2 is bracketed by green probes. And translocation results in swaps of red and green with a fused yellow signal, highly specific. I can think of DFSP, dermatofibromatosarcoma protuberans, where you have a classical T1722 PDGFR COL1A1 translocation. And you can use a fusion probe for that. Break apart probes. This is used if only one gene in fusion is known. It can be bracketed by a red probe on one side and green on the other, forming a yellow signal. And translocations result in separation of the probes, and you won't see yellow. But it does not identify gene partner. Chromosomal microarray. This shows copy number variations. Genetic copy number losses or gains, relative ploidy level of DNA in sample, is compared to a reference sample/database. This is the baseline, as you can see here. And
improved resolution for microdeletions and duplications, approximately 30 kilobases, and no prior knowledge of chromosomal aberrations are necessary, unlike FISH. And you can use it on formalin-fixed paraffin-embedded tissue. Here is a chromosomal microarray, a case of melanoma with numerous gains and losses of segments of chromosomes, where these represent chromosomes. Here it represents gains compared to the baseline, and here it represents losses compared to the baseline. And there are two main types of microarrays. One is an array-based comparative genomic hybridization, ACGH in short. It just detects copy number changes. A microarray platform contains probes covering the whole genome, and tests and control DNA are labeled with different fluorescent dyes. Both hybridize on a DNA chip. A relative signal intensity for each probe is analyzed. 2 is single nucleotide polymorphism SNP chromosomal microarray, SNP-CMA. It detects copy number variation, loss of heterozygosity, uniparental disomy. It allows to detect copy neutral changes not detected by ACGH. It contains both representative probes from throughout the genome, non-polymorphic copy number probes, and SMP probes, and only test DNA is added to the array. And the relative signal intensity is compared to in silico, or digital reference, to determine gains and losses. PCR, or polymerase chain reaction, a targeted amplification of nucleic acids of interest. Many methods rely heavily on PCR. It's the foundation of majority of clinical testing. It's highly sensitive and specific, and allows production of large quantities of product from rare targets, allowing for analysis. Standard PCR exponentially amplifies target DNA sequence, can be analyzed by a variety of methods. Primers created to region of interest and provide specificity. Reverse transcriptase PCR exponentially amplifies target RNA sequence, the transcriptome instead of genome, and it uses reverse transcriptase to convert mRNA to cDNA, which can then be amplified by standard PCR. It's useful for evaluating gene expressions and fusion genes, as well as RNA-based viruses, because PCR is done on DNA, right? And so for RNA-based viruses, you want to convert that to DNA before amplifying that DNA. Real-time PCR allows for detection and quantification of target DNA. It's based on detection of fluorescence during PCR. The signal increases in proportion to the amount of product formed. And this is important, and this is used for CML, where you look for that Philadelphia chromosome 922 and how much of it is there after the treatment. Sequencing, identifying the nucleotide order in nucleic acids. First generation sequencing usually refers to Sanger sequencing. It's based on DNA chain termination. It creates fragments of different lengths which can be separated by gel electrophoresis. Methodology is largely outdated. Next generation, second generation sequencing, NGS, a massive parallel read analysis of multiple genes simultaneously, physically separated in a small space, starting with a single DNA or cDNA fragment at each location and recorded in real time. You have increased sensitivity, specificity, and economics. It's the principal driver of genomic revolution and accessibility of clinical genetic analysis. And general steps are library preparation, clonal amplification, sequence analysis. Several different technologies exist with Illumina and iron torrent, Thermo Fisher, comprising the largest market share. Illumina, this generates sequence by reversible dye termination, a sequence by synthesis, and ion torrent, that generates sequence by measuring changes in pH when H is released from incorporated bases, ion semiconductor base. Regardless, this methodology utilizes extensive computational processing and bioinformatics involving base calling, read alignment, variant identification, and variant annotation. Sequencing depth versus coverage. Depth refers to the number of times a particular nucleotide is read during the sequencing process. Higher sequencing depth increases the confidence in calling a variant at a specific location. Coverage. the proportion of the genome or targeted region that has been sequenced at least once. Third generation sequencing. The techniques for a single molecule analysis without clonal amplification, mostly research use at present. Applications of NGS beyond genomics. There's RNA-seq, which is transcriptomics, and it identifies and enumerates many or most of the RNAs expressed in a sample, and methylomics. Methylomics is more prominently used in neuropathology. And so DNA methylation analysis, this is like genome-wide interrogation of DNA methylation signatures in conjunction with machine learning methods, and has allowed for the creation of the clinical grade classifiers, most prominently in CNS and soft tissue tumors. Tumor DNA methylation profiling has led to the identification of new entities and the consolidation of morphologically disparate cancers into biologically coherent entities, and it will likely progressively become mainstream in the future. I know for when I was on my neuropath rotation, we would tune into the NIH video stream, and there'd be samples sent all over the world to the NIH, which would do methylomics for free, and they would be able to classify the tumor based on the methylation signature, which was pretty amazing. So the methods of detecting DNA methylation, bisulfite conversion method, this is the most common method. Sodium bisulfite causes chemical conversion of unmethylated cytosines to uracil, which is then replaced by thymine during PCR. So C to U to T, and the ratio of cytosine to thymine at each potential conversion site is measured. Non-bisulfite dependent methods. Restriction endonuclease digestion. DNA incubated with methylation-specific enzymes, digested products are analyzed, and affinity enrichment. Methylated DNA is purified via immunoprecipitation, and the products are hybridized via array or NGS. When should I order molecular testing? Well, the main reason to order molecular testing, or any testing for that reason, is if it will change management of the patient. The main ways it can impact management are through altering diagnosis, or by offering a prognostic or predictive insight. So for diagnosis, as different tumors are being increasingly defined by specific molecular alterations, it's increasingly necessary to order confirmatory molecular testing. However, just because a confirmatory molecular test exists doesn't mean that it is necessary for diagnosis. Generally, Dr. Schaberg thinks testing is unnecessary when the diagnosis is reasonably certain based on other studies, including morphology. For example, if there is a retroperitoneal adipocytic tumor that looks classic for well-differentiated liposarcoma, ordering FISH for MDM2 amplification is likely an unnecessary cost and effort. But if it's a fatty tumor with equivocal morphology, MDM2 testing is likely required. That said, some untrusting but savvy clinicians may request it in all cases. Prognostic, predictive. So some genetic changes are prognostic and stratify patient outcomes, but do not determine therapy, and or predictive of if someone's cancer will likely respond to a given therapy. For example, if you have a BRAF V600 mutation in melanoma, you predict a beneficial response to a specific inhibitor like vemurafenib. These are both excellent reasons to get molecular testing, but should ideally be initiated by the clinical team who know if it is necessary for management. Which lesion should I test? If a stage 4 cancer patient is considered for molecularly informed therapy, a frequent issue is whether to analyze the primary tumor or a metastasis. There's no single easy answer, and sample availability and logistics often prevail. For example, if a patient has a lung mass and a liver mass, biopsying a liver mass is easier technically. It can often be done under ultrasound as opposed to CT, and it can potentially also help document metastasis. In the absence of therapy, primary tumors and metastasis show mostly the same driver gene mutations. Therapy often accelerate clonal divergence among the primary and subsequent metastasis. So basically do what you can with what you have. Practicalities often prevail. When heterogeneity is suspected, liquid biopsies for circulating tumor DNA or circulating tumor cells in the peripheral blood may reflect the clinical situation better, presumably integrating tumor status from a variety of sites and reflecting a composite mutational landscape. How much tissue is necessary for molecular? This is very important for us. because a clinician orders for molecular, and then if we don't know the criterion and send it for NGS and it comes back after time is wasted, it comes back and there's no definitive molecular results because tissue is inadequate, then time is wasted, and that's not fair for the patient. So minimum percent of tumor content needed depends on sensitivity of method for testing. So when in doubt, check for the desired test. But many state a sensitivity of 5% variant allele frequency, which corresponds to 10% tumor content, assuming heterozygosity. Notably, this is of all DNA nuclei present in this specimen, including inflammatory cells. There are a lot of other cells present. Macro or micro dissection can allow for tumor enrichment. FISH often requires fewer cells, but often still requires at least 20 identifiable tumor cells, and ideally more. What test should I order? There's no easy single answer. It really depends on what you are looking for, as there is no universal test. That said, generally, Dr. Schaberg uses the following tests in the following circumstances. IHC, as he's tried to highlight throughout, some molecular changes can be interrogated via IHC, particularly with next-gen stains like H3G34W for giant cell tumor bone. IHC is relatively fast, cheap, and easy to interpret, so this is always my first go-to test, if available, applicable. FISH, best for simple gene amplification translocation questions where you need limited information and would appreciate it fast. For example, that MDM2 amplification for a tumor you're suspecting is a liposarcoma. For example, he often gets this diagnostically for MDM2 amplification, as we just mentioned, and EWSR1 translocations in straightforward cases, where I'm pretty sure the diagnosis going in and just he needs a quick confirmation. For translocations, if the partner matters or if I'm not certain of the diagnosis, he often gets an RNA fusion panel. RNA NGS or RT-PCR fusion panel, it looks at a set selection of common RNA fusion transcripts. Best for cases that seem like they have a high likelihood of a fusion protein from a translocation, but where there are several possibilities. So a single FISH study is not ideal. Common scenarios where I get this, sarcomas and salivary gland tumors that seem like they have a high likelihood of having an underlying fusion, but where I am perhaps I'm not absolutely sure of the partners, particularly on a small biopsy where I may have fewer morphologic clues. Actually, we had a case in our institution recently of a secretory carcinoma, the salivary gland, and the biggest differential was actinic cell carcinoma. So, they have both different translocations. One is ETV6 NTRK 3, as well as actinic cell is the NR4A3 associated translocation. So we're still pending that. So NGS panel, best for looking for specific DNA mutations, currently based on the testing panel selected, although the technology can be used to query more things. Most commonly employed in the prognostic predicative setting, particularly looking for druggable targets, but can be useful diagnostically, particularly on small biopsies. If you look for MGMT hypermethylation for brain tumors like glioblastoma, you would be eligible for temozolomide, for instance. For example, diagnostically as well, Dr. Schaberg has had small GI spindle cell neoplasm biopsies that he thought were inflammatory fibroid polyps, but where scant tissue made this definitively diagnosing this challenging. In this case, inflammatory fibroid polyps to have a PDGFRA mutation helps support the diagnosis. The optimal panels of genes to target by NGS is controversial and evolving. Many panels focus on hotspots, regions within the genome that are commonly mutated, giving rise to specific clinically significant phenotypes. The middle ground between single gene and whole exome sequencing. It's thought to be most cost effective for now, and you may have different panels for different types of tumors, like for solid versus hematologic, based on mutation frequency, and often include commonly mutated tumor suppressors, oncogenes like TP53, genes mutated in specific neoplasms, assists in diagnosis and subclassifications, and genes targeted for targeted therapy, for like EGFR, you can do erlotinib, and BRAF, you can do vemurafenib. For the future, as sequencing and computation becomes faster and less expensive, it is possible that comprehensive genomic and transcriptomic sequencing will become standard. For example, Caris currently offers a single product with whole exome and whole transcriptome sequencing and tumor-relevant protein biomarkers. Additional resources, this is for your reference, and he recommends, in his limited experience, diagnostic pathology, molecular oncology. Okay. All right. Well, thank you so much for tuning in, and we will see you next time. Bye.